Replication. We all have the superpower to replicate. Well, more specifically, our cells have this power. Also, there are rules when it comes to replication, and the first affect the codes for our body, the DNA. First, meet the helicase. This enzyme unzips the genes.
That's what she said!
Once this enzyme unzips the DNA, the template strand, or the original strand, will serve as, well, the template for the daughter strand to form. Now with the DNA in this chopsticks form, a new enzyme comes around, DNA polymerase. This enzyme adds new nitrogen bases of the template strand. Keep in mind that two new DNA strands will be formed, as there were two template strands when the helicase unwinds the DNA. Although this may seem straightforward, a team of dedicated scientists that spend their lives looking at micropictures of DNA found photos to challenge the previous model of replication. This group decided to compare their pictures to the previous established model. Whereas originally the polymerases were thought to always remain at the bottom of the helicase, the newer information made it known that this was wrong. Instead, one polymerase was off to the side of the helicase, making a strand off to the side while the other strand was formed going down.
Transcription. Transcription is the process of DNA creating RNA. It's the first step in making proteins, and as we have learned from our first semester, proteins are very important in everything we do. They can catalyze reactions, contract muscles, become hormones, package DNA, and the most important task of doing much, much more. First, we need some DNA. The double helix structure of the DNA needs to be unwound by the helicase. As the helicase moves into the DNA, separating the strands, two strands are formed from the DNA. There is a sense strand and the antisense strand. The RNA polymerase helps find matching pairs for the newly forming RNA strand, using the antisense strand as a template. The DNA is closed as the RNA polymerase runs through the DNA. The end result is a newly formed RNA strand that uses the same nitrogen bases as the DNA strand that it copies, but for thymine, which is replaced with uracil. The RNA returns to ribosomes after the DNA strand sends a signal to the RNA polymerase to tell it to stop.
Translation. Translation begins with messenger RNA passing through a ribosome. As it goes for every 3 bases, which is a codon, the codon triggers a corresponding transfer RNA, which carries an amino acid to go through the ribosome and drop off the correct amino acid to form the polypeptide chain, as a result of the codon pairing it with the bases in the transfer RNA or the anticodon. At this point, the transfer RNA leaves, goes to the cytoplasm, and picks up another amino acid, restarting the process. As the ribosome moves along the messenger RNA, the polypeptide chain continues to grow longer and will eventually stop when it reaches the stop codon. The polypeptide chain is then released and will eventually become a protein. At that point, messenger RNA can repeat the process or be broken down after.
Connections to multiple units. Leukemia is a cancer caused by a mutation in DNA. It causes an overproduction of white blood cells, which oftentimes leads to a tumor. During replication, there is a malfunction in the genes that is supposed to stop mitosis in white blood cells. Cyclin E and A are proteins that regulate the different phases of the cell cycle. Cyclin E is responsible for repairing cells for DNA replication and lasts from growth 1 to growth 2. Cyclin A activates DNA replication and lasts from synthesis to mitosis. Proteins are essential in DNA replication and life. DNA holds the blueprints for proteins, and only through replication will a proper protein structure form. Proteins can then aid in more replication or be used for a variety of uses. RNA versus DNA. The origin of life considers RNA as the first way genetic code was stored, and later DNA took its place. The ribosome shows this by having RNA, whereas the nucleus has DNA. RNA can act as both genetic blueprint and an enzyme which allows for RNA to replicate without the need for other enzymes. CRISPR is a process of replacing DNA with other codons of DNA. To find certain sections of DNA, RNA is used as the matching pair for the DNA. Once located, CRISPR removes the nitrogen bases and replaces them with the correct bases. This allows scientists to change the genetic information of any living thing.